times that a cell can divide. Now I'd like to start with the basic structure of an animal cell. Now basically an animal cell contains some basic components, which are the nucleus, the cell membrane, the endoplasmic reticulum, and so on. But the main part of a cell is the nucleus. And the nucleus contains the DNA, which is responsible for protein synthesis, and it also controls all the functions of the cell. Now, the DNA that's present in the nucleus is present in the form of chromosomes. Now, let's take a closer look at these chromosomes. This is the basic structure of two chromosomes. Okay, now chromosomes are paired thread-like structures. This is one thread, and they're present in pairs. So there are two of them that are present in pairs. Okay, now basically they have a double helix structure. This structure that they're present in is called the double helix structure. Now towards the ends of the chromosomes, there are regions called telomeres. Now basically these telomeres are regions of repetitive DNA, which basically means that the same DNA that's present here is present towards the ends too, and this is to prevent the loss of DNA. Basically, these telomeres make sure that the chromosomes don't fuse with the neighboring chromosomes, and in the process, no DNA is also lost. They make sure of that. Now let's look at what happens to these telomeres when a cell divides. Now basically, cell division in cells, it takes place through the process of mitosis. Now mitosis is basically the basic process of cell division in which a parent cell divides to form two daughter cells. Now these two daughter cells are basically clones of the parent cell, which means that the DNA basically has to be cloned to form these daughter cells. Now in order for this DNA to be cloned, enzymes are responsible for it. So these enzymes are basically biochemical substances that are present in our body. So these enzymes, they can basically clone most of the chromosome, but they cannot clone the ends of the chromosome. Okay, not all of it. So basically, a part of the telomeres are left behind. They cannot be duplicated by the enzymes. So because of that, the telomeres that are present in the daughter cells are actually shorter than the telomeres that were present in the original parent cell, because the enzymes couldn't copy the whole thing. All right. Now let's look at a hypothetical situation. Imagine if telomeres did not exist in the DNA. Now what would happen? Basically, first of all, DNA would be lost because telomeres are responsible for the prevention of loss of DNA. And also, basically, if a cell didn't have telomeres in it and the DNA is copied, then the copy of the DNA wouldn't actually be a copy because a lot of mutations would be introduced, which means the genes in the DNA would change form and it would be very unstable. This situation is called genomic instability. Now basically, telomeres prevent the genomic instability from occurring. So that's why they're necessary. Now basically, if each time a cell divides, the telomeres shorten, then eventually a point will come where the telomeres are too short, so the cell won't be able to divide anymore, right? So at this point, the cell cannot divide anymore because if the telomeres are absent, then as I've told you, DNA would be lost and there would be genomic instability created. Hence, at some point, the cells have to stop dividing, alright? So this point at which the cell stops dividing because the telomeres are too short or they've reached their critical minimum length is called the Hayflick limit, alright? Now let's look at how the Hayflick limit was first discovered. Now basically, the Hayflick limit was discovered by an anatomist called Leonard Hayflick in 1961. Now basically, he proved the Hayflick limit through an experiment. He basically took three different cultures, alright? Cultures are growths of cells. So in one culture, he took male fibroblasts, human male fibroblasts. Fibroblasts are basically cells of the connective tissue, like bone or blood or something like that. So he took human male fibroblasts in one culture, which had divided around 40 times, female fibroblasts in another, which had divided around 10 times, and a mixture of the two. Now after some time, he noticed that in the mixture of the culture, the female fibroblasts had divided rapidly, but the male fibroblasts had been completely eliminated. Now the reason for this was that because the male fibroblasts had divided around 40 times already, they had reached their Hayflick limit and their telomeres had become too short for them to divide anymore. So because of that, they began to die through a process called apoptosis, which is basically programmed cell death. So they began to die, and that's how they were completely eliminated. And the blue ones, which are the male fibroblasts, were completely eliminated. And the culture only consisted of female fibroblasts. Okay? So that's how Leonard Hayflick proved the Hayflick limit. Now, although the Hayflick limit seems pretty simple and straightforward, there are actually loads of applications for it in real life. One is cellular aging. Basically, Leonard Hayflick identified 3 phases in a cell's life. One is when the cell is first created. Second phase is basically the growth of the cell, when the cell can rapidly grow. Leonard Hayflick actually identified this phase as the one of luxuriant growth. Now the third one is the period of senescence, which basically means the cell has reached its period of senescence, has reached its Hayflick limit, and because of this, it has began to die, and through the process of apoptosis. Okay. Now, scientists have also realized that cellular aging is directly related to the physical aging that's caused in the human body. So basically, since they're directly correlated, what happens is the Hayflick limit practically explains the mortality of human beings, why human beings cannot live forever. And that's how the physical changes are brought about in the human body due to age. Alright? Now the last thing that the Hayflick limit explains is cancer. Now cancer is basically the abnormal continuous division of cells, which means that cells divide continuously, alright, and that's when cancer is created. Alright, now the reason why these cancerous cells divide continuously is because they have an enzyme called telomerase, okay? Now this telomerase, it basically allows the telomeres to continuously divide without shortening. It basically maintains their length. Alright, so it makes sure that the cell can divide as much as it wants and the telomeres won't shorten at all. Alright, so that's basically what happens in cancerous cells. So there are 2 ways of preventing this, treating this disease or preventing it. The first one is by introducing telomerase inhibitors into the cancerous cells. Now what happens is these telomerase inhibitors, they inhibit the production of telomerase. So they stop the production of it, and because of that, the telomere length would not be maintained, and so the telomeres would decrease in length, and so the cancerous cells would start to behave like normal cells, and so the disease would eventually sort of decrease. Alright? The second method of treatment or prevention is by injecting telomerase activators into the cells of the immune system. What these do is that they activate the cells of the immune system, they activate the telomerase in them, so that the telomeres in them, they maintain their length, and so these immune cells can divide as much as they want, and so a lot of immune cells are created, which strengthens the immune system. Because of this, the immune system is capable of fighting off the cancer or preventing it in the first place. So with this, I come to the conclusion of the video. I really hope you've understood what the Hayflick limit is all about and how important and useful it is in real life. So with this, I sign off. Goodbye, and thank you for watching the video.
Safe.